Well, thank you to the organizers for this really wonderful event. It's been great to, meet people that I've known for some time and also some people I've never seen before, and also learn about all of the exciting work you're doing in your labs. So thank you. So today I'll tell you about one small area of research in my group that is focused on developing chemical biology tools. So we work at the interface of chemistry and biology, and I'm going to try to move around. If it becomes too distracting, tell me. Yeah, so we work at the interface of, I would say, chemistry, biology, and sometimes physics, wherein we try to employ tools from all of those disciplines to really try and dig into biological phenomena in an interest of advancing human health. I would say at the core of everything my group studies is this question of molecular interaction. We try to understand what drives proteins to interact with molecules in the cell, with other proteins, and other less common biomolecules, including DNA and lipids. And what drives our interest in these interactions is that we want to be able to mimic those interactions, to alter them or disrupt them in disease contexts. Okay. So two main areas in my lab, one primarily focused on discovery, another focused on applications. And you can imagine that our discovery processes can lead to development of new tools, and those tools can now be used to probe new biology in a variety of ways. So today I'll just talk about one of the stories on the application aspect from, I would say, the team building tools. So when you think about drugs and the kinds of molecules we can design drugs for, I would say in the traditional context is a protein that has some defined activity for which we can design a small molecule that is going to disrupt that activity, right? And that's great. That's led to a number of drugs targeting many classes of proteins. These are all enzymes that have a defined activity. What is excluded are all of the enzymes even that have other function that drive disease, or proteins that don't have this defined enzymatic activities. So how do we target those? Those remain part of the undruggable proteome, right? And there are quite a few of those. And in the last few years, a new set of therapeutic modality has emerged that doesn't require inhibiting a particular activity of an enzyme, but instead just allows you to lower or reduce the levels of that protein in the cell. And these are the heterobifunctional molecules in which you can have your protein of interest bind to a molecule that is linked to another molecule recruiting an E3 ligase. That E3 ligase is going to polyubiquitinate your protein of interest, mark it for degradation, and is then degraded by the proteasome. And that has expanded the druggable proteome in humans. But what remains still excluded are those proteins for which I would say that in these therapeutic modalities, you still need a ligand that binds to your protein of interest. And all of the PROTACs that are being used in clinical trials today or advancing to the clinic all are designed based on small molecules that were once drugs but are not effective inhibitors anymore. And so the enzymes, the proteins that are excluded are the ones that are not enzymes and ones that don't have this defined binding site for a small molecule. And that includes scaffolding proteins, transcription factors, and a wide variety of other proteins that still form part of the undruggable proteome. But what is common to all of these proteins is that they all engage in protein-protein interactions. So they do have interfaces to make interactions with other molecules in the cell. Now, can we leverage whatever information we have about what drives those interactions to now design ligands that would target those proteins, conjugate those to E3 recruiting elements, and then degrade those undruggable proteins? And one application for us is in the area of disorders affecting hemoglobin. Hemoglobin is one of the most important proteins in humans. It is responsible for oxygen transport to our organs and tissues. And because of its importance, gene deletions or mutations that affect hemoglobin can have a lot of adverse health effects. So I'm showing here two examples, beta thalassemia and sickle cell disease. In beta thalassemia, you have deletions in one of the subunits of hemoglobin, the beta subunit. And in sickle cell disease, you have a point mutation converting a glutamic acid to valine, introducing a hydrophobic patch on that protein that then leads to polymerization, aggregation in red blood cells in hypoxic conditions. And these disorders affect, I would say they're together are some of the most common genetic disorders in the world, affecting millions of people, predominantly in sub-Saharan Africa and in India. So there are a number of ways to treat sickle cell disease. I would say many of those therapies are present in the Western world and not really affecting the large millions of people around the world with sickle cell disease. And what's been known since the 80s is that expression of an alternate type of hemoglobin, known as fetal hemoglobin, can offer protection in sickle cell disease. In this study, I'm just showing that production of as little as 8% fetal hemoglobin can extend the probability of survival in patients with sickle cell disease by over 20 years. This is a form of hemoglobin that we all have. We produce it as fetuses, but it is silenced within the first year of life. And what offers this protection by fetal hemoglobin is the fact that it holds onto oxygen a little more tightly, and so those red cells remain mostly oxygenated even in hypoxic conditions. And after decades of studies, including genome-wide association studies, one key factor in the cell emerged as one of the most critical regulators of fetal hemoglobin levels, and that's this transcription factor known as BCL11A. And some of the classic work establishing the role of BCL11A was produced by Stu Orkin, work out of Stu Orkin's lab, in which they took a sickle cell disease mouse model, crossbred it with one that had BCL11A conditional and knockout. And you can see here in the peripheral blood smear, those are, I would say, classic of sickle cell disease patients where you have these sickle-shaped red blood cells. And when you have BCL11A knocked out, not only do you see more of those red blood cells, because again, one of the symptoms is anemia, you can see that these cells are quite normal. They don't have any sickling. And when you look into the cells for fetal hemoglobin levels, you can see that there is a drastic increase in the amount of fetal hemoglobin in these cells. Okay, because of that, there's been, I would say, a wide search for molecules that might increase fetal hemoglobin. Unfortunately, molecules that exist today do not target any of the key regulators of fetal hemoglobin. And one of those molecules that has emerged in, screening efforts is hydroxyurea. It's the only FDA-approved treatment for inducing fetal hemoglobin in patients today. But there are some limitations with this. For one, it is not universally effective. There are many patients who just don't respond to this treatment. And also, we don't really know how this molecule works, yet we give it to patients because sometimes it does increase fetal hemoglobin levels. The other strategy that is relatively new is through gene therapy, gene editing. In many cases, what they're actually doing is silencing or deleting the gene encoding BCL11A. And this therapy is quite effective at increasing the levels of fetal hemoglobin, but there are some concerns with genetic editing. And I would say key for us is the cost. Again, I mentioned that many of those patients with sickle cell disease reside in sub-Saharan Africa, and in India, they don't have access to the level of care that one would need to undergo gene editing. And in spite of this, there's, a lot of excitement, I would say, in the field due to these kinds of therapies. And whenever it is successful, it makes the news. What doesn't make the news is how grueling this is, right? So I'm just showing you here an abstract from one of these clinical trials in the process that patients undergo in order to get this kind of therapy, okay? So first you have to collect their hematopoietic stem cells via apheresis. Those cells are then edited ex vivo in highly specialized facilities, and a large number of them don't survive. And whatever is remaining at the end is then transplanted back into patients, and after engraftment, those patients begin to produce higher than normal levels of fetal hemoglobin. But before even transplantation, patients undergo myeloablative conditioning with chemo agents. And in some patients, that has led to cancer many years later, right? So this is not something that is offered to anyone who is not among the sickest of sickle cell disease patients. Okay, so given that we know BCL11A's role in inducing fetal hemoglobin, and its validation in those gene editing studies as being a way to develop therapies for sickle cell disease, why don't we create molecules that target this protein, right? So that's what my lab is trying to do. We're trying to create effective therapies for sickle cell disease by directly targeting those factors that are involved in inducing fetal hemoglobin levels. And we have programs that are focused on ligand discovery, delivery, and targeted protein degradation. Okay, so BCL11A has been known for a while, yet there are no conventional drugs that target it. Why is that? Well, it's one of the traditional undruggable proteins. There are no active sites to target. The full-length protein is really difficult to make, so even entering it into screens is something that's quite challenging. And then it has a specificity issue. There is another protein for which it shares a high sequence identity that has important roles in neuronal development. And so you would like to create ligands that is specific for BCL11A over that parallel. And so when I talk about the protein being difficult to make, this plot is just showing the disorder probability of the protein. Everything above this red line is predicted to be highly unstructured. And if you're not used to seeing plots like this is an AlphaFold prediction of what it looks like, and it's essentially gibberish, right? So working with something like this and trying to find a nice small molecule that is going to bind to a pocket and be specific for BCL11A over its parallel is highly unlikely. And so how do we work with this? Well, there are some regions of structural order in here, and those regions actually share more than, I'm showing you 69 overall, but in those structurally ordered regions, the identity approaches 100% between the two paralogs. But we're biochemists, and we have to start somewhere, okay? So in order to find ligands for BCL11A, we've created fragments of the protein that are focused around these ordered regions, but are extended into the disordered regions, and our goal is to identify ligands that would prefer binding to the disordered regions of BCL11A over the structured fragments. Okay, so when you're starting to think of disordered proteins and going after them with ligands that would be specific, you're really limited in your choice of ligands. But we do know that those regions of BCL11A engage in protein-protein interactions, and so we've considered using nanobodies, which are fragments of antibodies, as potential ligands for BCL11A. And so I'll just quickly walk you through some of our data showing how we selected targets. This is a story on just one of those fragments of BCL11A. Again, we're working to target our nanobody ligand to that disordered region of BCL11A. We entered it into a screen, so we do screening using either phage display or use surface display to identify nanobodies that are selected for BCL11A. Hits are then evolved into better binders. We then validate those targets, those hits using in vitro methods, showing again the specificity of BCL11A over its paralog. We then use quantitative biophysical measurements to determine the interaction, the strength of the interaction between those ligands and BCL11A, and structural approaches to eventually optimize the design of our nanobodies. Okay. So BCL11A is a transcription factor. It's in the nucleus, and we're going after it with protein-based ligands. They're not going to cross the barrier of the cell membrane on their own. And to functionalize them for cell penetration, we've been using these cell-permeant miniature proteins that can transport protein cargo into the cell. And by just creating a fusion with one nanobody with this delivery vehicle called ZF5.3, you can see that the affinity for that fragment of BCL11A is not significantly altered. We can now take this and just incubate it with cells. So I'm showing you images here from confocal microscopy where we're just looking at the ability of this protein now fused to the delivery vehicle, shown here in red, to enter these erythroid progenitor cells. And the protein gets into cells and co-localizes to the nucleus by evidence of this magenta spots in our confocal images and the merged versions of them. We've looked at the dose dependence of this, so from 1 micromolar to 10 micromolar of this protein gets into cells and accumulates in the nucleus. We've looked at the time dependence in as little as 30 minutes. You can see cell entry and nuclear stabilization. We've looked at this over 24 hours. It appears that the protein continues to accumulate in the nucleus. And one thing that was key to us was to ensure that this nanobody fragment, nanobody that was selected using only a fragment of BCL11A, still has affinity for the full-length protein. and that we can deliver sufficient amounts to engage with full-length BCL11A. And that's what this experiment is showing, wherein we can deliver the nanobody and use it to pull down BCL11A in the elution. Yeah. Okay, so this is good, and getting that nanobody in and interacting with BCL11A does not alter the function, does not change fetal hemoglobin levels, but because we're chemical biologists, we can use it as a way to now recruit an E3 ligase to degrade this transcription factor. And that's what we did using two distinct E3 ligases here. One works better than the other, and I'll just talk about this data using this E3 ligase called SPOP, wherein within a few hours after incubating with cells, you can see much of BCL11A has been depleted in these cells. We've done this multiple times, looked at the kinetics. This appears to be quite stable, even longer, 72 hours, and in later experiments, up to 5 days. BCL11A levels remain low in these cells. We've done some key control experiments in which we omit the nanobody from the construct, and you see that leads to loss of the degradation. And when you inhibit the proteasome by adding this inhibitor, MG132, you see that there is no change in BCL11A levels. So this is all happening via proteasomal degradation. So we then looked at the resultant impact on fetal hemoglobin levels. And in order to do this, we'll have to differentiate these cells from a more stem cell-like state into mature erythrocytes. This experiment is just looking at protein levels. When we do this versus untreated, you see that BCL11A levels are low even during differentiation after we've added the degrader. And excitingly, we begin to see higher than normal levels of fetal hemoglobin. We've looked at the transcripts of this, either showing here the actual levels or the full change, and you can see that there is like a 2- to 3-fold change in gamma hemoglobin levels in these patients. And when you stain these cells for fetal hemoglobin, again, there is a 2- to 3-fold increase in fetal hemoglobin in these hemoglobin-positive cells. Okay, so all of that was done in a cell line. We're trying to target actual hematopoietic stem cells, and so we went and got patient-derived CD34 cells and did this experiment there. And again, similar to what we saw before, we can efficiently degrade BCL11A, see the resultant increase in fetal hemoglobin levels using Western blots and also by following the mRNA of the gamma hemoglobin. We've done this in multiple donors. We see the same effect. Again, if you start with very low levels of fetal hemoglobin, the increase is modest, but the full change is the same. And so this is not donor-specific. We've also done some housekeeping studies where we're just looking at the fitness when we've treated our erythroid progenitor cells with this degrader. You can see that there is a reduction in the proliferation, but this has been attributed to loss of BCL11A as opposed to additional toxicity from our degrader. And we've looked at things like viability and the effect on differentiation, and we see no difference between treated and untreated. And so what we've been able to do is identify a nanobody that is specific for a disordered protein or disordered fragment of BCL11A. We've been able to functionalize this ligand for cell penetration and degradation by adding an E3 recruiting element, leading to BCL11A loss. And this loss then leads to fetal hemoglobin induction, as has been achieved in patients via gene editing. And what is really exciting about this is that the levels of fetal hemoglobin increase we're seeing, if we can achieve that in patients, those will be at levels that are curative. But another aspect of this is that this gives us a tool that we can now use to probe the role of this particular transcription factor in other disease contexts, including reported roles in promoting metastasis in triple-negative breast cancer. And we're also excited about the fact that we might now have a means to target those other undruggable proteins for which ligand identification is still a problem. So with that, I'd like to thank the people who did all of this work. One phenomenal postdoc, Fang Shen, members of my group that have worked on protein modulation, other members of my group are here. We're fortunate to collaborate with Stu Orkin and his postdoc, Ge Zhang, and I'm grateful to these sources for funding, and I'll be happy to take questions at the end if there's any time remaining. Thank you.